A signal transduction pathway is a series of steps by which a signal is converted into a specific cellular response. It involves the binding of a signal, also known as the primary messenger, to the signal transducer, usually located on the plasma membrane, which activates a primary effector, triggering the release or production of second messengers, which in turn activates secondary effectors, and eventually generates various cellular responses through the cascade. In this video, I'm going to cover 7 main types of signal receptors. G-protein coupled receptors, abbreviated as GPCRs, are plasma membrane receptors that work with the help of the G-protein, which is a heterotrimeric protein consists of alpha, beta, and gamma subunits. The alpha subunit serves as a binary switch that is inactive when bound to GDP and active when bound to GTP. The hormone epinephrine, also known as adrenaline, produced by the adrenal medulla as part of the fight-or-flight response, binds to a type of GPCR known as beta-adrenergic receptors, located on the plasma membrane of muscle, liver, and adipose tissue to mediate changes in fuel metabolism. The beta-adrenergic receptor, abbreviated as BAR, is an integral protein with 7 hydrophobic helical regions that span the plasma membrane 7 times. The binding of epinephrine to beta-adrenergic receptor promotes a conformational change in the receptor's intracellular domain that affects its interaction with an associated stimulatory G-protein, abbreviated as GS, causing the GDP bound to alpha subunit to be replaced by GTP, activating the G-protein. The beta and gamma subunits of G-protein dissociate as dimers, and the alpha subunit with its bound GTP moves in the plane of the membrane from the receptor to the primary effector adenylyl cyclase, abbreviated as AC, which is another integral protein of the plasma membrane that converts ATP to cyclic AMP, or CAMP. Cyclic AMP serves as a second messenger that allosterically activates CAMP-dependent protein kinase, also known as protein kinase A or PKA, which is made of 2 identical regulatory subunits and 2 identical catalytic subunits. Normally, the regulatory subunits block and inhibits the active sites of the catalytic subunits. When cytosolic concentration of CAMP increases, 4 molecules of CAMP bind to 2 regulatory subunits of PKA, causing the dissociation of the 2 regulatory subunits and revealing the substrate binding site on the 2 catalytic subunits, which in turn catalyzes the phosphorylation of specific serine and threonine residues of targeted proteins. As I mentioned in my video on glycogen metabolism, both epinephrine and glucagon can promote glycogen breakdown in the liver, a process known as glycogenolysis. Epinephrine triggers an enzyme cascade that amplifies the original signal, increasing blood glucose concentration by 100,000-fold. In many cell types, the catalytic subunit of PKA can also move into the nucleus, where it phosphorylates the CAMP response element binding protein, abbreviated as CREB, which alters the expression of specific genes regulated by CAMP. Cell signaling is often confined to a specific region of the cell by adapter proteins, also known as scaffolding proteins, which are non-catalytic proteins that hold together other protein molecules that function in concert. In the case of epinephrine signaling, the scaffolding protein aka P5, which stands for A kinase anchoring proteins 5, attached to the membrane through palmitoyl and phosphoinositol triphosphate, and binds to beta-adrenergic receptor adenylyl cyclase PKA as well as phosphoprotein phosphatase to bring all the enzymes in close proximity. There are 4 main mechanisms that terminate the epinephrine signal transduction pathway. The first mechanism involves desensitization of beta-adrenergic receptor to an inactive conformation. As mentioned earlier, the binding of epinephrine to the beta-adrenergic receptor triggers the dissociation of beta and gamma subunits as dimers from the activated alpha subunit. The beta-gamma dimer recruits the beta-adrenergic receptor kinase, abbreviated as BARC, which phosphorylates several serine residues near carboxyl terminus of the receptor on the cytoplasmic side of the plasma membrane. The phosphorylation of the receptor creates a binding site of the protein beta-arrestin, abbreviated as BAR. Receptor bound to beta-arrestin enters the cell by endocytosis, and beta-arrestin dissociates. In this state, the receptors are inaccessible to epinephrine and therefore inactive. The receptor is eventually dephosphorylated and returned to the cell surface. The second termination mechanism involves intrinsic GTPase activity on the G-protein, which catalyzes the hydrolysis of GTP bound to the alpha subunit. Conversion of the bound GTP to GDP favors the return of the alpha subunit to inactive conformation, in which it binds beta and gamma subunits. thus decreasing the activity of adenylyl cyclase. The GTPase activity can be enhanced by GTPase activator proteins, abbreviated as GAP, allowing fine-tuning of the epinephrine pathway. The intrinsic GTPase activity of the G protein on the surface of intestinal epithelial cells can be inhibited by the cholera toxin, which catalyzes the transfer of ADP ribose from NAD+ to a critical arginine residue on the alpha subunit of G protein, blocking the GTPase activity. As a result, G protein is permanently active, causing continuous activation of adenylyl cyclase of intestinal epithelial cells. Chronically high CAMP and active PKA results in the efflux of sodium chloride and leads to severe osmotic imbalance. The third termination mechanism involves phosphodiesterase 3 or 4, which hydrolyzes the second messenger CAMP to the inactive 5' AMP. Finally, a decrease in the cytosolic concentration of CAMP would decrease the amount of active PKA. Therefore, more targeted proteins will return to its dephosphorylated state by the action of protein phosphatase. As I covered in my video on glycogen metabolism, protein phosphatase 1, abbreviated as PP1, reverses the effect of various kinases. Epinephrine can have different effects on different tissues or cell types. In tissues with alpha-2 adrenergic receptors, abbreviated as alpha-AR, which is coupled to the inhibitory G protein GI, epinephrine acts by inhibiting adenylyl cyclase, thus lowering CAMP and suppressing protein phosphorylation, eliciting other fight-or-flight responses such as vasoconstriction and decreasing gastrointestinal activities. Besides epinephrine, many other signals also bind to G protein coupled receptor and use CAMP as second messengers, such as odorants, taste sense, dopamine, histamine, etc. Humans' ability to detect light also relies on G protein coupled receptor, though through a different pathway. Recall in animal physiology, light is detected by photoreceptors known as rod cells, located in the retina. The special part about this signal transduction pathway is that the rods are depolarized by default and actually hyperpolarized when activated by light, which is opposite of action potential and other transduction pathways. First, the light strikes the G protein coupled receptor known as rhodopsin, abbreviated as RH, which is made of the protein component opsin and the chromophore retinol, which is derived from vitamin A1. Light converts 11-cis retinol to O-trans retinol, causing a conformational change that activates the rhodopsin molecule. The activated rhodopsin then interacts with a special class of G protein known as transducin. Rhodopsin catalyzes the replacement of GDP by GTP, thereby activating transducin. Similar to the G protein of beta-adrenergic receptor, the beta and gamma subunits of transducin dissociate. The activated alpha subunit of transducin then activates the primary effector, cyclic GMP phosphodiesterase, abbreviated as PDE, which converts cyclic GMP to 5' GMP. Under dark environment, cGMP binds and activates cation channels, causing an influx of sodium and calcium cations, which depolarize the rod cells. When PDE is activated by light, the concentration of cGMP reduces, causing the cation channels to close, leading to hyperpolarization of rod cells. Phototransduction is terminated very quickly, which shares some mechanisms with epinephrine termination pathway. Similar to beta-adrenergic receptor, rhodopsin can also be desensitized to an inactive conformation. Once the cation channels are closed, continued action of sodium-calcium exchanger reduces the cytosolic concentration of calcium. Under low calcium concentration, rhodopsin kinase, abbreviated as RK, phosphorylates the carboxyl terminal domain of rhodopsin, which creates a binding site for arrestin 1, abbreviated as ARR. Arrestin 1 is homologous to beta-arrestin of the epinephrine pathway. It sequesters rhodopsin, making it unable to interact with transducin. After a while, arrestin 1 eventually dissociates from rhodopsin. The O-trans retinol in transducin is replaced with 11-cis retinol, and the rhodopsin is ready for another phototransduction cycle. Similar to the G protein stimulated by epinephrine, transducin also contains intrinsic GTPase activity that hydrolyzes GTP to GDP, inactivating the alpha subunit of transducin and decreasing the activity of phosphodiesterase. Another recovery process involves restoring the cytoplasmic cGMP levels. A decrease in the cytosolic calcium concentration activates guanylyl cyclase, abbreviated as GC, converting GTP to cyclic GMP, which binds and activates the cation channels, causing the rod to depolarize again. Another broad class of G-protein coupled receptors utilizes phospholipase C as its primary effector, such as acetylcholine, angiotensin, oxytocin, etc. Acetylcholine that utilizes G-protein coupled receptor mainly function in the parasympathetic nervous system, promoting rest and digest physiological responses. First, the neurotransmitter acetylcholine binds to the muscarinic acetylcholine receptor, abbreviated as MACHR, activating the G-protein GQ by replacing GDP with GTP. The beta and gamma subunit of GQ dissociate, while the alpha subunit of GTP activates the primary effector phospholipase C, abbreviated as PLC. Active PLC cleaves the membrane phospholipid phosphoinositol 4,5-bisphosphate, abbreviated as PIP2, into inositol 1,4,5-triphosphate, abbreviated as IP3, as well as diacylglycerol, abbreviated as DAG. Inositol triphosphate is water-soluble and diffuses from the plasma membrane to the endoplasmic reticulum, where it binds to specific IP3-gated calcium channels, causing them to open. Calcium cation diffuses out of the endoplasmic reticulum, increasing its cytosolic concentration. Both calcium and diacylglycerol serve as second messengers and activate protein kinase C, abbreviated as PKC. There are several isozymes of PKC, each with a characteristic tissue distribution, target protein specificity, and role. The PKCs activated by acetylcholine phosphorylase target enzymes and generally play a role in the parasympathetic nervous system, triggering rest and digest responses such as stimulation of digestive juices and gastrointestinal motility. The significant rise of cytosolic concentration of calcium cation is also detected by calmodulin, abbreviated as CAM, which is an acidic protein that is part of the calcium calmodulin-dependent kinase, abbreviated as CAM kinase. When calmodulin binds calcium cations, it undergoes a conformational change that activates CAM kinase. The kinase then phosphorylates target enzymes, such as myosin light chain kinases that trigger the contraction of smooth muscle. The level of calcium ions typically oscillates with a period of few seconds. A second major class of signal receptor is receptor tyrosine kinases, abbreviated as RTK, which is responsible for the signal transduction pathways of insulin and many other growth factors. The hormone insulin, produced by the pancreas when blood glucose level rises, is the primary anabolic hormone that promotes glucose uptake, glycogen synthesis, and fatty acid synthesis. First, insulin binds to two identical alpha subunits of insulin receptor, abbreviated as INSR, on the extracellular phase, which triggers each of the two transmembrane beta subunits to phosphorylase specific tyrosine residues on the carboxy terminal on the cytosolic side. This autophosphorylation opens up the active site so that the enzyme can phosphorylate tyrosine residues of IRS1, which stands for insulin receptor substrate 1. From IRS1, the insulin signal transduction branches into two separate pathways. In the cytosolic pathway, the enzyme phosphoinositide 3 kinase, abbreviated as PI3K, binds the phosphorylated IRS1 through PI3K's SH2 domain. The activated PI3K then converts the membrane lipid phosphatidylinositol 4,5-bisphosphate, abbreviated as PIP2, to phosphatidylinositol 3,4,5-triphosphate, abbreviated as PIP3. Notice that PI3K shares the same substrate as phospholipase C from the acetylcholine pathway. However, phospholipase C cleaves PIP2 into inositol triphosphate and diacylglycerol, while PI3K phosphorylates PIP2 into PIP3. PIP3 in turn binds and activates protein kinase B, abbreviated as PKB, through phosphorylation by another protein kinase PDK1, which stands for phosphoinositide-dependent kinase 1. As I mentioned in my previous video on glycogen metabolism, PKB phosphorylates and inactivates glycogen synthase kinase 3, preventing it from inactivating glycogen synthase. Inactivated glycogen synthase ultimately promotes glycogen synthesis, also known as glycogenesis. IRS1 also branches into another pathway that affects gene expression. First, the phosphorylated tyrosine residue of IRS1 binds to the SH2 domain of the protein GRB2, which is an adapter protein that also binds to the proline residues of the protein SOS through the SH3 domain. SOS replaces the GDP bound to the G protein RAS with GTP, thus activating it. GTP-bound RAS then activates RAF1, the first of the three protein kinases that form a phosphorylation cascade. RAF1 phosphorylates the second protein kinase MEK, which in turn phosphorylates the third protein kinase ERK. The activated ERK then enters the nucleus and phosphorylates other transcription factors to activate the transcription of proteins essential for cell division. In this case, insulin acts as a growth factor. The proteins of RAF, MEK, and ERK are members of 3 larger families. ERK is in the MAPK family, which stands for mitogen-activated protein kinases. Mitogens are extracellular signals that induce mitosis and cytokinesis. Subsequently, the protein kinase MEK belongs to the MAPKK family, which stands for MAPK kinase, and RAF1 belongs to the MAPKK family, which stands for MAPKK kinase. The MAPK cascades also mediate signaling initiated by other growth factors, including platelet-derived growth factor and epidermal growth factor. Another type of receptor that uses tyrosine kinase is the cytokine receptors. Cytokines are small signaling molecules that function in developmental regulation and immunity. An example of cytokine is erythropoietin, produced by kidney, which regulates the development of red blood cells. First, erythropoietin binds to the erythropoietin receptor, abbreviated as EPOR, causing the receptor to dimerize. The receptor dimer then binds and activates the soluble cytosolic tyrosine protein kinase JAK, which stands for Janus kinase. Notice that the difference between cytokine receptor and receptor tyrosine kinase is that cytokine receptors lack intrinsic tyrosine kinase activity, while receptor tyrosine kinase does have intrinsic tyrosine kinase activity. The activated JAK then phosphorylates several tyrosine residues in the cytoplasmic domain of erythropoietin receptor. The phosphorylated erythropoietin receptor can activate 2 different signaling pathways. In one pathway, the adapter protein SHC binds the phosphorylated tyrosine residue of erythropoietin receptor, while also binding to GRB2, triggering the MAPK cascade that increases gene expression for cell division of erythrocytes, also known as red blood cells. In the second pathway, the phosphorylated tyrosine residue binds to the SH2 domain of the STAT5 protein, causing 2 STAT protein to dimerize, exposing a nuclear localization sequence that targets the dimer for transport into the nucleus. In the nucleus, STAT5 turns on the expression of genes that increase the red blood cell production. The fourth major class of signal receptors is receptor guanylyl cyclase, abbreviated as RGC, which are receptors with intrinsic guanylyl cyclase that converts GTP to the second messenger cyclic GMP, or cGMP. In the kidney, receptor guanylyl cyclase is activated by the peptide hormone atrial natriuretic factor, abbreviated as ANF, which is released by cells in the cardiac atrium when the heart is stretched by increased blood volume. ANF activates the receptor guanylyl cyclase known as ANF receptor, located in the collecting ducts of kidney. Once bound to ANF, the intrinsic guanylyl cyclase in the receptor, abbreviated as GC, converts GTP into cyclic GMP or cGMP. The rise of cGMP increases the renal secretion of sodium ion and consequently water, driven by the change in osmotic pressure, thereby lowering blood volume, returning it to homeostasis. Regulation of blood pressure and volume has been covered in my video on homeostasis. In the vascular smooth muscles, the rise of cGMP activates cGMP-dependent protein kinase, also known as protein kinase G, or PKG, which triggers an enzyme phosphorylation cascade that leads to vasodilation, or relaxation of blood vessels, increasing blood flow while further decreasing blood pressure. Another type of signal that acts through receptor guanylyl cyclase is the gaseous local regulator nitric oxide. Nitric oxide is sufficiently nonpolar to cross the plasma membrane and binds to a soluble receptor guanylyl cyclase found in the cytosol, abbreviated as sRGC. Nitric oxide binds to a heme group attached to a soluble receptor, causing the intrinsic guanylyl cyclase to convert GTP to cyclic GMP, which activates protein kinase G and also results in vasodilation of various smooth muscles, including gastrointestinal tract and the erectile tissues. Similar to the termination of epinephrine pathway, which degrades cAMP to inactive 5' AMP, cyclic GMP can also be degraded to the inactive 5' GMP by the enzyme phosphodiesterase 5, abbreviated as PDE5. The drug sildenafil under the brand name Viagra inhibits PDE5 found in the blood vessels of the penis, therefore causing cyclic GMP to remain elevated, resulting in prolonged vasodilation, effectively treating erectile dysfunction. Notice that so far I've covered protein kinases A, B, C, and G. To recap, protein kinase A or PKA is activated by cyclic AMP in the epinephrine pathway. Protein kinase B or PKB is activated by phosphoinositol triphosphate in the insulin pathway. Protein kinase C or PKC is activated by both calcium cation and diacylglycerol in the acetylcholine pathway. Protein kinase G or PKG is activated by cyclic GMP in the adrenal-neurouretic pathway as well as the nitric oxide pathway. As mentioned earlier, the neurotransmitter acetylcholine functions in the parasympathetic nervous system, promoting rest and digest physiological responses. Another major function of acetylcholine is to stimulate muscle contraction at the neuromuscular junction between the motor neuron and muscle fiber. The binding of acetylcholine to the nicotinic acetylcholine receptor, abbreviated as NACHR, located on the muscle fiber, triggered the opening of the receptor ion channel, allowing the passage of only sodium and calcium cations. The influx of sodium ions causes the membrane to depolarize, triggering an action potential that propagates along the plasma membrane down the transfer tubules, promoting the release of calcium cation from the sarcoplasmic reticulum. An increase in the concentration of calcium ions in the cytosol of the muscle fiber triggers cycles of muscle contraction. More details about muscle contraction have been covered in my animal physiology video on the muscular system. In my previous video on action potential, I talked about how different neurotransmitters can bind and activate specific ionotropic receptors, causing either local depolarization or local hyperpolarization, known as excitatory postsynaptic potentials, or EPSPs, and inhibitory postsynaptic potentials, or IPSPs, respectively. The combination of EPSPs and IPSPs ultimately determine whether the membrane potential will reach the threshold for action potential or not. The major excitatory neurotransmitter is glutamate, which binds to ionotropic glutamate receptors, abbreviated as IGLURs, in the postsynaptic neuron, permitting the influx of sodium and calcium cations, leading to local depolarization or EPSP. On the other hand, the major inhibitory neurotransmitter is GABA, which stands for gamma-aminobutyric acid, which binds to ionotropic GABA receptor, abbreviated as GABA-AR, permitting the influx of chloroanions, leading to local polarization or IPSP. The sixth type of receptor is integrins, which carry signal bidirectionally between the cytosol and the extracellular matrix. All integrins have one alpha and one beta subunit, each with a short cytoplasmic extension, a single transmembrane helix, and a large extracellular domain with ligand binding site. For the outside-in signaling, extracellular ligands such as collagen or fibrinogen interact with the ligand binding site of integrin and straightens the extracellular domain, causing the cytosolic tails of alpha and beta subunits to move apart and altering their interactions with intracellular proteins such as talin, alpha-actinin, vinculin, paxillin, and others to conduct the signal inward. For the inside-out signaling, contact of the integrin by talins and other proteins also causes the integrin to straighten, increasing its affinity for extracellular binding partners. The bidirectional signaling of integrin function and coagulation, immunity, cell division and differentiation, and tumor invasion. The last type of signal receptors are nuclear hormone receptors. Lipid-soluble hormones, including steroid hormones, thyroid hormones, and melatonin, travel in the blood and carrier proteins. They diffuse across the plasma membrane of target cells and bind to specific receptor proteins, triggering a conformational change in the receptor protein and converting it to a hormone response element, abbreviated as HRE. HREs are capable of interacting with specific regulatory sequences in DNA to alter gene expression.